Good morning, good afternoon, and good evening. Welcome back to Chapter 6, Cellular Respiration. Remember, our goal is to make ATP from a food molecule, and in this case, the food molecule we're talking about is a molecule of sugar, a molecule of glucose. As a quick recap, we started with a molecule of sugar, a 6-carbon molecule that we cut in half. I got two 3-carbon molecules called pyruvic acid. In addition to the pyruvic acid, I got 2 ATPs and 2 NADHs. I'm trying to get as much energy out of this sugar molecule as I possibly can. That energy is going to be in the form of ATP. And here's 2. Remember that NADH, we will deal with all of the NADHs that we make at the end, step 3. So all the ones we make, we send to step 3. ATP is cash in my pocket. NADHs are tips on the credit card. I've got 2 tips on the card here. I got $2 to put in my pocket, so those are gone. So I'm left with my 2 pyruvates. I take my 2 pyruvates to step 1.5. When I take them to step 1.5, I'm going to cut one of those carbons off and release it as carbon dioxide. In place of where that carbon was, I'm going to put a molecule called coenzyme A. Remember that enzymes have a job to do, and the job that this enzyme is going to do is it's going to help these 2 carbons get into the Krebs cycle, to get into the citric acid cycle. Because I have 2 pyruvic acids, I will make 2 carbon dioxides, cut 1 off of each. I will therefore make 2 acetyl-CoAs, and as an energy molecule, I will make 2 of those NADHs, 1 for each. Remember, where do the NADHs go? The NADHs go to the last step, step 3. So that's 2 more tips on the card. 1, 2, 3, 4. The job of the coenzyme A is to get these 2 carbons into step 2, which is the citric acid cycle, sometimes called the Krebs cycle. So here is my acetyl-CoA. Those 2 carbons we're going to attach to a 4-carbon molecule that was already present in the mitochondria. Remember, everything after step 1.5 takes place in the mitochondria. Step 1.5 takes place in the matrix of the mitochondria, so does the citric acid cycle. 4-carbon molecule plus 2 carbons equals 6. The reason why it's called the citric acid cycle is because the first stable product of the citric acid cycle is citric acid. The coenzyme A comes off, and it's going to get reused, and it'll get recycled again the next time the cell will break apart a sugar molecule. That 6-carbon molecule, I'm going to cut a carbon off and release its carbon dioxide. Now I have a 5-carbon molecule. That 5-carbon molecule, I'm going to cut another carbon off, release it as carbon dioxide, and now I have a 4-carbon molecule. This 4-carbon molecule isn't that one. I know it looks the same, but again, remember the invisible hydrogens and oxygens? I have to convert it into that one, because this is a cycle. So I have to end with what I started with. At various stages throughout this process, and we're not going to say where, and it doesn't really matter, it's just that it does happen. I will get 3 NADHs, so that's 3 more tips on the card. I'll get 1 ATP, 1 more dollar in my pocket, and 1 FADH2. And we said that FADH2 looks a lot like NADH. It's similar, it's just smaller. Now, just like we did last time, how many of these acetyl-CoAs do I have? At the end of step 1.5, how many did I make? I made 2. So how many times do I do Krebs? I do it once for each. So I do it twice. I do this whole process twice. So at the end of the citric acid cycle, because I do the whole process twice, I don't get 1 FADH2, I get 2. I don't get 1 ATP, I get 2. And I don't get 3 NADHs, I get 6. Because I do the whole process twice. Once for each. Now, that means Each time I send one of these acetyl-CoAs in, I release their carbons as carbon dioxide. I lost 2 carbons here. Remember, I started with 6. I lost 2 of them here, so now I'm down to 4. I lose 2 when I do it the first time. Now I'm down to 2. The last 2 come in, I run it again, and there goes my last 2 carbons. All 6 of my carbons are released as carbon dioxide. All of them that came into my glucose, they all get released as carbon dioxide. 2 here and 4 here, because I run this process not just once, but I run it twice. So 1, 2, 3, 4. The Krebs cycle. And this is what we just said, that the 2 carbons are cut off and releases carbon dioxide. I get 1 ATP, 1 FADH2, and 3 NADHs per acetyl-CoA. And what did we just say a second ago? How many of those do we have? Well, at the end of step 1.5, I had 2 of them, so I don't do this process once, I did it twice. I make that 4-carbon molecule again. I remake it at the end of the cycle so that I can do what? So I can do it again. And again, this happens in the matrix. This is a really, really stripped down version showing what the process is. My acetyl-CoA enters the cycle, the CoA comes off. Ultimately, the 2 carbons will be cut off and releases carbon dioxide. At various stages in here, I'll get 3 NADHs, an ATP, and an FADH2. But because how many acetyl-CoAs do I have at the end of step 1.5? I have 2 of them. I do this whole process not once, but twice. So I get 4 carbon dioxides, 6 NADHs, 2 ATPs, and 2 FADH2s. We've now made it to step 3. We've now made it to the end of the night, and now you get to cash out. The real question is, how many tips on the card did you earn tonight? Well, at the moment, you have $4 in your pocket. You have $4 in your pocket because you made 2 here and 2 there. That's your $4 that you have. Next, we have to add up all the NADHs and FADH2s we made because those are tips on the credit card. There's 2 there, 2 there, 6 here. 2 plus 2 plus 6 is 10 NADHs, and then 2 of these FADH2s. I have a total of 12 tips on the credit card that now I get to cash out at the end. And the end is the electron transport chain. And as the name implies, we're going to transport electrons. We're going to take those electrons that we ripped off of the glucose and put onto the credit card, put onto NAD+, turning it into NADH. We're going to pull those electrons off, and we're going to allow them to drop down a series of chemical stair steps. Let's just kind of slinky. Just like a slinky going down a set of stairs, it releases a tiny bit of energy at each step. As these electrons are passed from one molecule to the next, they release a little bit of energy, and that energy is used to make ATP. The very last step, the very last thing that takes the electrons away, so more electrons can come down the steps, is this stuff that we call oxygen. I will combine the oxygens plus those electrons, the hydrogens that I pulled off to make this stuff that we call water. This is the electron transport chain. Each NADH carries enough energy in it to make 3 ATP, and FADH2 carries enough to make 2. So how many NADHs did I make? Well, I made 10. What's 10 times 3? That's 30. At the end of the night, I had $4 before I walked up to the cash register, before I walked up to my manager and said, okay, I'm ready to go home. He cashed out all of those 10 NADH tips and gave you $30. Then you had 2 of these smaller tips called FADH2. He cashed both of those out, and they were only worth $2 each. So 2 times 2 is 4. So at the end of the night, your manager hands you $34 to go along with the 4 you already had in your pocket. So you made a total of $38 by weight and tables. All of this process, this electron transport chain process, takes place across the inner membrane of the mitochondria. Remember what the mitochondria looks like. Kind of looks a little bit like a kidney bean. We have a highly folded inner membrane. Step 1.5 and step 2 both happen in this center area called the matrix. Step 3 is going to happen across this inner membrane. We're going to move stuff from inside the matrix to this narrow area between them, what's called the intermembrane space. What does inter mean? Inter means between. So the intermembrane space literally means the space between the membranes. We're going to move stuff from the matrix to the space between the membranes.